This will be a walkthrough of the AQA GCSE Biology 2023 Higher Tier Paper 1. Question 1.4. Scientists have extracted chemical A from the deadly nightshade plant. Chemical A can be used as a painkiller. Table 1 shows information about where chemical A is found. The scientists usually extract chemical A from the berries of the deadly nightshade plant. Suggest one reason why berries are used instead of leaves or roots. So looking at the table, we can see the levels of each of the three parts of the plant. They want us to focus on berries. So if we look at the value for berries is 0.7, which is the lowest out of the three. Remember that chemical A can be used as a painkiller. So ideally, we would want to get as much of chemical A from the plant as possible. However, we don't know how much chemical A can be used to treat. So maybe the dosage that comes from the berries, in terms of its mass of chemical A, is sufficient. If there was anything further, then maybe it could be harmful to the person. So we can say berries have the lowest mass because it has the appropriate dosage to not cause harm to the person. What else could you say? Well, let's think about the other parts of the plant. The berries are only going to be there for fruit. That's its only purpose. The leaves are there to carry out photosynthesis. So they have a function. The roots are there for anchorage and to absorb water and mineral ions from the soil. So if we were to take the leaves away from the plant, it would stop photosynthesis, so the plant would be harmed. If we were to take the roots away, then it wouldn't be able to absorb water, it wouldn't be supported in the ground, and then it would damage and harm the plant. So the berries can be taken because removing the berries won't cause any harm to the plant. So that's another possible answer there. The final thing you want to think about is how can you obtain the chemical A itself? Trying to get the chemical A from roots is going to be difficult. Trying to get the chemical A from the leaves would be easier than the roots, but still would be a bit tricky. Whereas getting the chemical A from the berries should be the easiest. So it would be the easiest to extract. Question 1.8. There are many online reports making claims about the effects of chemical A. Some of these reports are biased. Suggest one reason why a report making claims about the effects of chemical A may be biased. Remember what biased means. It means favoring a certain situation, condition. So, why would someone writing a report be biased towards the effects of this chemical and what they do? Well, maybe they are benefiting from it in some way. If someone writes a report and they want to say that something is very, very good and has a good use, they may be wanting to sell it. So they would like to make a profit. On the other side of it, maybe somebody else might be trying to sell chemical A too. So they don't want the person that is selling it also to gain from it. So it's to prevent competitors gaining money. Question 2.8. Explain the effect of a partly blocked coronary artery on the human body. 6 marks. So let's remind ourselves now, coronary artery, what is its job? it's supposed to supply oxygen to the heart muscle. So it's not allowing blood to be pumped, but it's helping the heart to do that. So what would be the issue if this blood vessel is partly blocked? Now, it didn't say fully blocked. Now, if we were to talk about fully blocked, we should then be thinking no oxygen would be supplied. So we've got to try and remember now that if it's partly, we should be trying to use words like less, slower, reduced. Remember, the question is asking us to explain the effect on the human body. So we can't just think about the heart itself. We need to think about the rest of the body. Now, remember that the rest of the body now is really just going to be cells and tissues, organs. So I want to think about what would be the effect there. Well, reminding ourselves about the job of the heart again is to pump blood to the rest of the body. So if we want to pump blood to the rest of the body, that means that we want to supply oxygen to these cells, to these tissues. Why would we want to do that? It's for respiration. So let's just jot down job of heart, to pump blood to the body. What we can also think though is that the heart is a double circulatory system. So it's not just going to pump blood to the body, it's also going to pump blood to the lungs too. And then we've just reminded ourselves that the body is cells and tissues. Now, if it's going to pump blood to the body, what do we want the blood to contain? We want the blood to contain oxygen. So this blood should have O2. If we want the blood to be pumped to the lungs, we want that blood to contain CO2, carbon dioxide, because we want to get rid of it through exhalation. So, what would be the reason for the oxygen going to the cells and tissues? Again, it's respiration. For what purpose? What does respiration allow for us to have? It allows us to have energy, provides energy. So, cells, tissues gain oxygen for aerobic respiration. And that is to release energy. Okay, so we have now reminded ourselves the actual jobs here. So all we can do now is we can think, if it's partly blocked, what are the issues? So first thing then, so as we said, the job of the coronary artery is to supply oxygen to the heart muscle, so there'll be less blood supplied to the heart muscle. What's the consequence of that now? Less oxygen supplied to the heart muscle cells. Okay, now, what are the muscle cells supposed to do? Contract, and that's supposed to pump the blood. So if they are getting less oxygen, there'll be less contraction, so there'll be less blood pumped around the body. And then the consequence of less blood pumped around the body, again similar to the point above that, there'll be less oxygen supplied to tissues. The consequence of that means there'll be less aerobic respiration. And that causes less energy to be released. Remember, if there is less aerobic respiration, that should mean automatically there'll be more anaerobic respiration. And one of the main differences between aerobic and anaerobic respiration is the amount of energy that's transferred. There'll always be less energy transferred in anaerobic respiration compared to aerobic respiration. Now, what are the effects of that on the human body? If you have less energy released, how would you experience that? What would you see? Well, the person would just be tired. What we can then say, link back to what we were talking about here about anaerobic respiration. If there's anaerobic respiration, there would then be muscle fatigue. So, more anaerobic respiration leads to muscle fatigue. Now, we have to say the reason why there's going to be muscle fatigue with this anaerobic respiration. And it's the result of lactic acid production. So more anaerobic respiration causes muscle fatigue due to production of lactic acid. And those are our points. If you look at what we've done now, we have, for each of our points now, we have sort of explained a little bit further now what are the effects. Question 3.2. During a food poisoning outbreak, scientists identified the farm where the food came from. The farmer had not vaccinated the farm animals against Salmonella bacteria. The food poisoning outbreak could have been prevented if the farm animals had been vaccinated. Explain how. The immune systems of animals respond to a vaccination. The immune response in farm animals prevents an outbreak of food poisoning in humans. 4 marks. So what's the key point from this question now? They've went and told us the specific type of pathogen, Salmonella bacteria. And we remember that bacteria produce toxins and they reproduce rapidly. Next key point in this question is vaccination. What is a vaccine? It's a dead or weak version of a pathogen. Now, whenever we're thinking about vaccination, we should always have this graph in our mind. Time and antibody concentration. And this is known as your primary and secondary immune response. So first thing that will happen is you will be injected with a vaccine, and that will cause a response in the body leading to production of antibodies. Eventually, the antibodies are going to help to destroy the pathogen, which is why the antibody concentration decreases. But then you're exposed to a live version of the pathogen, and the response will be faster and greater because the immune system has recognized what the antigen looks like, what the pathogen is, and then it will remain in the blood for longer. So, here's our response. Once we introduce the vaccine, the white blood cells will produce antibodies. So vaccine causes white blood cells to produce antibodies. Antibodies do. Well, they bind to a specific antigen. In this case, we should assume that the vaccination should be some dead or weak version of Salmonella. So, the antibodies are going to bind to the antigens of Salmonella, and they bind specifically to that. And what is going to happen when they bind? They're going to cause them to be destroyed. Okay, so we have discussed now what will be the response. So 2 points for that. Let's look at the second bullet point now. The immune response in farm animals prevents an outbreak of food poisoning in humans. Okay, well, if a human is going to get food poisoning, that's most likely because the food itself has been contaminated with this pathogen. But if we look at what's happened here now, our antibodies have destroyed the pathogen, Salmonella bacteria. And this is happening in the animal. Remember, this is not happening in the human because the animals have been vaccinated, as it says in the question. So, what does that mean for humans now? When the humans eat, the food should not contain any pathogen, any Salmonella, because they've been destroyed. So, we should just say then, humans not affected, as no pathogens, especially Salmonella here, are on the food. Okay? Remember, they have been killed already in the animal. Now, let's just lastly, because we haven't made it clear first, let's just lastly clarify that these 2 points here, this will be in our first part of our graph, which is this primary response. What we want to make sure that we mention in our answer finally is what happens in the secondary response. So, in the secondary response, if the farm animals were exposed to the live version of Salmonella, the antibodies would be produced rapidly and they would destroy this Salmonella quickly. So let's just write that. Question 3.4. The scientists incubated bacteria at 37 degrees Celsius. Students in school laboratories incubate bacteria at 25 degrees. Explain why scientists use 37, but scientists must use 25 degrees to incubate bacteria. Okay, so a certain temperature is restricted in schools for growing bacteria. So, why? What do we want to prevent? Every time that we're doing a culturing microorganism experiment, we're trying to avoid contamination and prevent the spread of harmful pathogens. So, bacteria like a warm condition. When it's nice and warm, they will grow more. So by keeping it at 25 degrees, you're preventing the growth of harmful pathogens. Okay, but why have it at that specific temperature of 37 degrees? Why not 40? Why not 42? What's special about 37 degrees? Well, what's special about that temperature is that it is the human body temperature. Now, why would a scientist want to incubate a bacteria at 37 degrees? Maybe they're interested in seeing how quickly a bacteria grows in a temperature that is relative to human body temperature. So, remember that we're dealing with a question that's linked to the previous question to do with salmonella. So what it probably would mean then is that the salmonella itself, this type of bacteria, grow best at 37 degrees. What is that term called when bacteria grow best or something is at its best? The term is the optimum. So their optimum temperature of growth is 37 degrees. Question 3.9. Antibiotics treat food poisoning because they kill salmonella bacteria inside the human body. Some antibiotics work because they damage the bacterial cell wall. The bacteria die because the cells burst. Explain why the cells burst. 3 marks. So the bacteria are in the human body, traveling around there. The antibiotic, when ingested, will damage the cell wall of the bacteria, and then eventually this bacteria die because the cells are burst. So let's think about a cell wall now of a bacterial cell. If that is disrupted, what does that mean now that the bacteria are going to be exposed to more? Well, if they're traveling around the human body, they're going to be traveling in the blood. And what is blood mostly made up from? It's mostly made up from plasma, which is mostly made up from water. So all around this bacterial cell will be water if it's traveling in the blood. Damage in the cell wall means that there'll be some holes or gaps appearing in the cell wall. So what does that mean is going to happen now? The water is going to enter the bacterial cell. So that should be our first mark now. We have to remember now that we have to explain ourselves. So how is the water going to enter the bacterial cell? Well, every time you start to think of water in biology, you should always be thinking about osmosis. So it's going to enter the cell by osmosis. Remember that the definition of osmosis is the movement of water molecules from a high concentration to a low concentration through a semi-permeable membrane. But some water can enter, but why is that going to cause the cell to burst? It must mean that there's so much water that the cell wall can't withstand the pressure that the water is providing. Question 4.2. Design an investigation to show the effect of different types of exercise on the heart rate of athletes. 6 marks. Now, there is no required practical for this question. This is just an indication of how well you understand doing investigations. So, what should you think about when you are trying to carry out an investigation? Your variables, your independent variable, your dependent variable, and your control variables. So, from reading just the sentence, which is our question, we should be able to straight away identify the independent variable and the dependent variable. Independent variable means change. So reading the question now, what is changing? It says different types of exercise. So we want to change the type of exercise. Dependent variable means measure. So what do we want to measure in the experiment? We want to see the heart rate of the athletes. So control variable, something that we want to keep the same. Well, we're seeing how the heart rate of athletes change based on the exercise. something about the athletes we want to try and keep the same. Now, we could keep the athletes the same, but we want to try and use as many athletes as possible. So we want to try and keep some things the same about the athletes. What could vary a lot, which could influence the heart rate? First thing is their age. So the age should be similar. Next thing that you want to try and keep the same, their sex. So you want to have all males or all females, for example. The next thing that you want to try and keep the same then is, are they healthy? Are they suffering from any health issues? So what you probably want to say is no health issues. So now that we figured out our variables, we just want to get it set up. So remember we were saying that we want to use athletes. So what we'd want to say then is we want to get a group of athletes. You can give a number if you wish, but just have a group of athletes. Remember, you want them to have a similar age. Group of athletes of similar age. Say same sex. And with no health issue. Remember, we're going to now change the type of exercise. So, I want to then write down, get athletes to perform a certain type of exercise. For example, jogging. Now, to see the effect on the heart rate, we can't just measure the heart rate once. We'd have to measure the heart rate before they do the activity and after they do the activity. So we just need to make that clear. Now, even though we're using similar age, same sex, and no health issues for this group of athletes, their heart rate at the beginning, before the activity has taken place, can still differ. So what we want to make sure that we do then, to enable us to compare it, is to find the increase in their heart rate. Once we measure the heart rate at the start and the end of the activity, we then want to find the increase in the heart rate. Okay. Then what we can do is we can say repeat investigation, but with a different activity. But we have to then think, if our athletes have done some jogging and then we have measured their heart rate, right after the activity their heart rate is going to be high. If we then immediately get them to do the next activity, it's going to be difficult to compare because their heart rate would have already been higher than at the start. So we want to say repeat the investigation with a different activity, and then what we can do is you can give another example, running. But what we want to say is ensure that the heart rate has dropped before starting the activity a second time. And there is a suitable investigation to allow us to compare the heart rates. Question 4.3. Anabolic steroids or drugs. Anabolic steroids increase muscle mass in humans, are banned in most competitive sports. Some athletes take anabolic steroids to improve their performance in sport. Explain how taking anabolic steroids could improve an athlete's performance. 2 marks. Well, what does an improvement in athlete's performance look like? It sounds like a very vague term there, so let's try and give an example of an improvement in an athlete's performance. Well, a perfect example is an athlete running faster. Because again, that improvement has got to be something that is better than before. So faster is an example. Then maybe lifting some weights, so they'll be stronger in that sense. Maybe throwing something further could be another example. But what's the reason why taking these steroids is going to cause this to happen? Well, it told us that the steroid is supposed to increase muscle mass. So there's a greater proportion of muscle in the athlete. Now, whether it's running faster, whether it's throwing something further, whether it's being stronger to cause something to be lifted up greater, it all requires the muscles. What are the muscles supposed to do during any of those activities? The muscles are going to contract. So if the person is running faster, that's got to mean that there are more muscle contractions. Question 5.3. During one stage of malaria infection, the malarial protists enter blood cells and cause them to burst. Explain why the bursting of red blood cells causes tiredness. Well, it's a similar situation to one of our previous questions. We're just talking about a cell bursting. But in this case of the malarial protist, it's not bursting because there's an increase in the water that's entered the cell. It's bursting because there's an increase in the amount of protist because they reproduce inside the red blood cell and eventually cause the cell to burst. But the question is different because now what we've got to do is you've got to link red blood cells to tiredness. Well, what's the function of a red blood cell? It's there to carry oxygen. So if there is bursting of red blood cells, then that's got to mean you have less red blood cells, so that means that there's less oxygen that's going to be carried around the body. However, tiredness, we've actually used this term already. The previous question, when it was asking us to explain the effect of a partly blocked coronary artery on the human body, well, what was the reason that we said causes someone to be tired? It's because there's less aerobic respiration. So there's more anaerobic respiration, and the difference between them is there's less energy transferred in anaerobic respiration compared to aerobic respiration. So that's what we just need to say. There'll be less energy released, and that's going to be due to more anaerobic respiration. Question 5.5. One drug for treating malaria prevents mitosis occurring in the malarial protist. The drug stops the synthesis of new DNA bases in the cell. Suggest how the drug prevents mitosis occurring. So, mitosis. Mitosis is cell division, and it involves chromosomes dividing, and the nucleus dividing, and then cytoplasm cell membrane splitting to form two new cells. However, mitosis does not involve DNA bases. So we can't really be thinking about something within mitosis being directly affected as a result of the bases. Well, remember that mitosis is part of the bigger picture, which is the cell cycle. So if this is mitosis, you have stage 1, stage 2, and stage 3. Stage 2 is mitosis. Stage 3 is known as cytokinesis, and that's where the cell membrane and cytoplasm divide. And stage 1 is simply known as cell growth. What happens during this first stage then? Two main things are happening. there's an increase in the number of your subcellular structures or organelles, and there is DNA replication. So let's look at what it says again. The drug prevents mitosis occurring in malarial protist. So stage 2 can't occur. The drug stops the synthesis of new DNA bases in the cell. So adding or making new bases. What's the drug going to do then? That means it's going to interfere with stage 1. And stage 1, we said, is DNA replication. So it stops DNA from replicating. Question 5.7. Different types of disease may interact. Scientists studied the incidence of malaria infections in children without disorder S and with disorder S. The incidence of malaria in children with disorder S was calculated as a percentage of the incidence in children without disorder S. Table 4 shows the results. Describe what the results in Table 4 show about the interaction between disorder S and malaria. 2 marks. So let's first look at the table and just describe it. So we can see that going down in the table, in our first column, the age is increasing, going from 2 to less than 4, all the way to more than 10. In our second column, we can see the calculated percentage incidence of malaria in children with disorder S, and we can see that it decreases, but when it gets to more than 10, it actually increases there. So what we can just say then, because we're describing our table, we should be able to just get a mark for describing, is as the ages go from 2 to 10, so increase from 2 to 10, the percentage incidence of malaria in children with disorder S decreases up until the age of more than 10, where it increases. That should be a clear first point because we have described the table. So as age increases, the percentage incidence of malaria in children with disorder S decreases. Now, I've already put a circle around this sentence because it's a very interesting thing that they've put separately on their own and felt it was necessary to tell us. So let's read it again and try and figure out what it means. The incidence of malaria in children with disorder S, so in this table, was calculated as a percentage of the incidence in children without disorder S. Remember that they're trying to study children with disorder S and without disorder S. So what this is telling us then really is that these percentages are those with disorder S, but then it's saying it as a percentage of those without disorder S. So that means then it's not a percentage of all the children, it's a percentage of those that do not have disorder S also. So that means that whatever is remaining should be people or children with malaria that do not have disorder S. So what we would need to do then is take this value away from 100, and then that should give us the value of children without disorder S that have malaria. So 100 minus 61 should be 31. 100 minus 63 should be 37. 100 minus 50 is 50. 100 minus 45 is 55. And 100 minus 73 should be 27. So what can we say now? Well, the people with malaria that do not have disorder S is actually increasing as we age. So, what is that actually telling us then? That's actually telling us then, because looking at interactions here, that if someone has malaria and someone has disorder S, they're actually less likely to develop malaria if they have disorder S. And that should be our next point. Question 6.7. Explain why the leaf discs move to the surface of the solution during the investigation. Well, let's look at the investigation first. A student investigated the effect of color of light on the rate of photosynthesis in leaves. So like we did before, we can identify by reading this sentence what's the independent variable and what's the dependent variable. So the effect of color of light. So that means that they must be changing the color of light and seeing what that does. It says on the rate of photosynthesis in leaves. So they want to measure the rate of photosynthesis somehow. Now this is directly then linked to your core practical on photosynthesis. However, they have slightly altered it. Instead of you having a beaker and a plant and you're measuring the number of bubbles or the volume of oxygen produced, they're then measuring, and we can see it from our results table here, the time taken for 10 leaf discs to reach the surface of the solution in seconds. So you can see that they have changed the color of light, so we were on the right lines for our independent variable here. But this time taken will be the true dependent variable, the true thing that's going to be measured. Now it said reach the surface of the solution, so that means that these discs are actually going to rise up in this beaker of sodium hydrogen carbonate solution. But the question they want us to answer is, why did the discs move? Well, if they're leaves, they're supposed to be photosynthesizing. For them to photosynthesize then, that means that they must be producing two things, glucose and oxygen, and that means that they must be using carbon dioxide and water. So what would be the reason why the leaf discs start to increase in terms of their position? Why would they start to rise? Well, they're not going to rise because the glucose is getting produced. Because the glucose would probably be a heavy molecule, because it's fairly large in comparison to the other things in the equation, and then that would cause them to stay where they are. But our oxygen is getting produced, and our oxygen is a gas. And we know that gases are very light or they are very dense. So they would like to rise up. Think about a balloon. When you fill up air into a balloon, the balloon rises. It's got to do with density. So the first thing that we should be making clear in our answer is that oxygen is being produced. And it's being produced during photosynthesis. Now, what will the oxygen do then? The oxygen, when produced, is going to cause the leaves to get pushed up because they are not very dense. There are two types of chlorophyll in leaves. Figure 10 shows the percentage of different wavelengths of light that the two types of chlorophyll absorb. You have chlorophyll A, dashed line, chlorophyll B, solid line. Table 6 shows the color of different wavelengths of light. So we've got a variety of different colors of our rainbow, and then for each one they've got a range of wavelengths that can be part of. Suggest the advantage to a plant of having two types of chlorophyll. Well, let's go back to the graph and let's study it a bit more because we didn't do it the first time. So we can see chlorophyll A. Chlorophyll A is present in this section, whereas chlorophyll B doesn't have much in that section there. Opposite occurs next. Chlorophyll B, a high amount there, but chlorophyll A is at a lower amount. Between 500 and 600, roughly, they're fairly low for both pigments. And then, just around 650, there is high amount of chlorophyll B, but a greater amount of chlorophyll A just to the right of it. So what it's telling us essentially then is the times in which chlorophyll A is at its maximum are at a different wavelength of light when chlorophyll B is at its maximum. Why is that useful to a plant? Well, if we take both of these chlorophylls to be in a plant, that essentially means that the range in which they can absorb light is going to be greater. If it was just chlorophyll B, then the range would be a bit shorter because you wouldn't have anything just around 500 or just before 500. You wouldn't have anything just after 600 because it would just be chlorophyll A. Conversely, you wouldn't have anything below 450 if there was no chlorophyll A. You wouldn't have anything just before 700 because there is no chlorophyll A. So the advantage is can absorb a greater wavelength range of light. (6.9) Table 5 is repeated below. So it's the same table, however we didn't analyze the result. So we can do that now. The leaf discs in this investigation are green. Explain the results in table 5 for blue light and for green light. Use data from figure 10 and table 6. So we're going to go through table 6 now, and then we can analyze the result there. But then we've also got to go back to figure 10, which was the graph. Got to analyze that a little bit more carefully now, so we can come up with an answer which is worth 4 marks. Now, first thing then is the table. So it says time taken for 10 leaf discs to reach the surface of the solution in seconds. So if you look when it's blue light, it takes 115 seconds for the disc to reach the surface. Green takes 831 and red takes 397. So the fastest we can clearly see then is blue and the slowest is green. Underneath the table, separate sentence is one of these notable sentences that we need to make a note of. The leaf discs in the investigation are green. So the leaf discs are the same color as the color which took the slowest. Explain the results for the blue and green light. So let's go back to green light now. Remember that they've given us a table, table 6, and it says we've got the color of light green there. Remember that this was the slowest. What's the reason? There is a wavelength of light 500 to 570. So we're looking at 500 to just before it reaches 600. So what do we notice? The wavelength of light is between 500 to 570. The color of the light is green. The percentage of light absorbed is the worst. So it's not absorbing any light. If something is not absorbing any light, then it means one of two things. It means the light is passing straight through it, or the light is being reflected. And in this case, it's going to be very tricky for us to say that the light is passing through the leaf. So we're going to say that the light is being reflected. So for the green light is being reflected. And what we just want to make sure of is that we can compare this to blue light also. So we can say that green light is being reflected the most. What other way do we know that? If you look at table 5, it's taking the longest time for the disc to reach the solution. Now, if it's taking the longest time for the disc to reach the solution, what does that tell us? Let's go back to that previous question that we answered, 6.7. We know that the oxygen is supposed to be produced in photosynthesis, and the oxygen, due to its low density, is going to push the disc upwards. So that must mean then, if the leaf discs are taking the slowest time or the longest time, it must mean that the rate of photosynthesis is the slowest. We then have to talk about the blue light. So let's do the same analysis now. Blue light, fastest, 115 seconds. Going back to our table 6, blue light has the range 450 to 499. Let's find that in the graph. 450 to 499, just before 500. What do we notice? Chlorophyll B absorbing the most. 450 though, is chlorophyll A also absorbing the most at that point too? So both chlorophyll A and chlorophyll B are absorbing light the most at that point. The leaf discs are taking the shortest time to reach the surface. The rate of photosynthesis must be the greatest.